I'm going to be telling you about my doctoral dissertation research on sexual differentiation of the brain. And it's obvious to most of us that men and women are different. And even in young children, there are often obvious differences in really simple things like toy choice between boys and girls. And you might think that things like preferences and toy choice are largely dependent on the environment in which the kid is raised and our expectations of what the kid should be interested in based on their gender. But even super progressive parents who try to raise their kids in a gender-neutral environment can sometimes be really kind of shocked and appalled at how their kids kind of naturally gravitate towards gender-biased toys and activities. But it turns out that monkeys do this too. So male monkeys prefer to play with trucks and cars, and female monkeys prefer to play with dolls. So how can this be? Like, as far as we know, monkey moms don't have expectations of what their monkey kids should be playing with. So gender is a uniquely human social construct. We've made these concepts up for ourselves. But sex differences in the brain are biological. And sex differences in the brain can determine things like preferences and behaviors. And so, considering how non-human primates have sex differences and preferences, it indicates that there is some kind of biological basis for what we see in kids. Sex differences in the brain control some of the most important behaviors that an animal or a human can display for its survival and reproductive success. So some examples of these behaviors are things like aggression, parental care, and sex, or reproductive behaviors. And there's sex differences in these behaviors across the animal kingdom. And it's really interesting from an evolutionary point of view to try to understand how these behaviors came about. So this is one really good reason to try to understand the basic biological differences that determine the male and female brain. Another super important reason to study sex differences in the brain, and the reason why I study sex differences in the brain, is because your sex is your number one risk factor for having a psychiatric or neurological disorder. You might have heard before that boys are more likely to have disorders like autism, ADHD, early onset schizophrenia compared to girls. And we consider these disorders neurodevelopmental because they show up early on when they're kids. Females are more likely to have disorders that occur post-puberty, such as depression, anxiety, eating disorders, and social phobias. So since there are such striking sex differences in the prevalence of these disorders, it's really super critical that we understand the basic biological differences between the male and female brain if we want to truly understand how these disorders arise. We need to do manipulations of the brain in order to infer causality. We cannot rely on correlation for these things. And that's why we turn to animal models. And most people who study sex differences in the brain use rodent models like mice and rats. The rodent genome is really similar to ours, and on a cellular and molecular level, their brains function pretty much the same way as ours do. Granted, the human brain is way more complex, and we have higher order structures that they don't have. But essentially, the rodent brain is kind of just like a simplified version of our brain. What's so good about using rodents to study sex differences is that they exhibit super robust differences in behavior that are quantifiable. And one example of a behavior that has sex differences in rodents is the behavior that you see here, sex behavior. So the male rat shows this mounting posture, and in response to the mounting, a female displays a lordosis response. And this is a 21 and over crowd, so I'm going to be showing you a lot of rat porn tonight. But I just want you to keep in mind that I don't care about rats having sex. And actually, I don't care about humans having sex either. The reason why we study sex differences in the brain, and the reason why we look at sex differences in behaviors like sex behavior, is basically as a readout to determine if a brain is masculinized or feminized. So, if we want to know if a brain is masculinized, we quantify male sex behavior. So just keep that in mind throughout the rest of the talk. So the sex differences that we see in rat behavior, rat and mouse behavior, are created by sex differences in brain structures. And these are also very robust and quantifiable in the rodent brain, making them a good model. And what makes them better than humans is that we're actually able to manipulate their brains, as you'll see. And in this way, we're able to determine what the actual molecular mechanisms are that create sex differences in the brain. Okay, so before sexual differentiation of the brain occurs, two other separate processes usually occur. The first process is called genetic sex determination, and this happens at conception when you receive your chromosomes from your parents. And in mammals, if you're a male, you get one X and one Y chromosome. And if you're a female, you get two X chromosomes, one X from your dad, one X from your mom. If you're a male, you have a gene on your Y chromosome called SRY. And SRY codes for a protein called testes determining factor. And it's this protein that changes the bipotential reproductive system and the bipotential gonad into a testicle. Once these testes are made, they begin secreting lots of different hormones, and these hormones cause the female primordial reproductive tract to disappear, and they encourage the development of the male reproductive tract. In the absence of SRY, the bipotential gonad typically develops into an ovary, and a lack of testicular hormones enables the development of the female reproductive tract and causes the primordial male reproductive tract to disappear. So maybe you've heard people say in the past that females are the default sex. And this is the reason why. It's because usually in the absence of hormones and in the absence of these genes on the Y chromosome, the reproductive tract will develop into the female phenotype. And typically the individual will develop as a female, a normal female. So following sexual differentiation of the gonads, sexual differentiation of the brain occurs. And in humans, this happens during the second trimester in utero. And in rodents, it happens just around the time of birth. And it happens during what we call a critical period. So a critical period is a time when the brain is sensitive to certain inputs. And it's only during this time frame that the brain can respond to a specific input, and it can't respond to this input either before or after the critical period. So the critical period for sexual differentiation of the brain just so happens to coincide with a huge surge in testosterone from the developing testes. So these levels of testosterone are as high as we see in an adult male. So it's just a huge level that the testicles are pumping out into the blood. So these hormones travel through the blood and they reach the brain. And high levels of hormone exposure in the bipotential brain is what causes brain masculinization. In females, there's super low levels of hormones during the critical period. And this allows for feminization of the brain. So just like you saw with gonadal sex determination, sexual differentiation of the brain has been referred to as the default developmental process. And this is because for many years, people studied this and they couldn't figure out what factors or hormones initiate the process of feminization of the brain. So people considered it the default, and they considered it sort of a passive developmental process. But the research that I did in grad school, which I'm going to tell you about, challenges this dogma, and it shows us that feminization of the brain is actually an extremely active process, and it's the active process of suppressing masculinization via epigenetic mechanisms. And I know that sounds complicated, but I'm going to break it down. So hormone exposure during this critical period causes lifelong masculinization of brain structures. This is essentially what creates structural sex differences in the brain. So sex differences in the brain can come in many different forms. So for instance, we can see differences in the size of a brain region between males and females. Sometimes a brain region is bigger in males, sometimes it's bigger in females. And then we also see sex differences on a cellular and molecular level. And so these can look like differences in the size and shape and connectivity of neurons and also other cells in the brain. And this is programmed by hormone exposure during this critical period. So hormone exposure during the critical period enables adult hormones to activate the male or female brains. In this case, I'm showing the male brain. So these males have been masculinized during the critical period, and then adult levels of testosterone enables them to show male-specific behavior, in this case, sex behavior, such that if we administer hormones during the critical period to a female, she will grow up to behave as a male. And what this video is showing is male sex behavior in a rat, except it's actually a female who was born a normal female. She's genetically female. She looks like a female on the outside. But we've given her a dose of hormones on the day that she was born. And this causes lifelong masculinization of the brain. Just ignore them trying to escape. Okay, so if you think about this, it's just crazy and amazing that such a brief, fleeting hormone exposure, basically like a blip in hormones in these animals, so early on in development, can have these profound changes in the animal's brain and behavior. So this is what really fascinated me during grad school, and it still fascinates me now. So the question that I really wanted to address with my doctoral dissertation research is how? How is this? How do hormones actually create sex differences in the brain? What's the mechanism for this? And then also, what makes sex differences permanent? So I've shown you that the hormone exposure during the critical period is fleeting and brief, and yet it causes permanent organization of the brain. And if you know anything about neuroscience or the brain, you probably have heard that the brain is considered plastic, and especially during development. So it's crazy that something can be programmed lifelong like this, especially something so important. So these are the questions that I wanted to address with my dissertation. And just around the time that I started grad school, epigenetics was starting to become a hot topic. And epigenetics is kind of like an umbrella term for several different molecular processes that control gene expression without actually affecting our DNA sequence. So one definition is that it's a type of cellular memory that enables experiences and environment to influence gene expression without changing DNA sequence. There are several different types of epigenetic processes, but the one that I studied is called DNA methylation. And in general, DNA methylation is associated with genes being turned off or silenced. So DNA methyltransferases, or DNMTs, these are the enzymes that methylate DNA. And methylated DNA stops gene expression in general. So high levels of DNMT activity cause genes to be shut off. If there's low levels of DNMT activity and low levels of DNA methylation, this allows for genes to be turned on. And various environmental signals and normal developmental processes influence which genes are turned on and turned off via DNA methylation. And DNA methylation is considered a long-term epigenetic modification. So although epigenetic marks can come and go, and they do, especially in the brain, they can be established early on and can essentially be permanent. And sometimes they can actually be heritable as well. So you can pass DNA methylation patterns onto your offspring in some cases. So I hypothesized that maybe a mechanism for how hormones are permanently masculinizing the brain during the critical period is to masculinize patterns of DNA methylation. So I wanted to quantify DNA methylation in the male and female brain to see if there was a difference and to see if hormones affected this difference. But since the brain is so complex, and some regions have really strong sex differences and other regions have no sex differences, I wanted to focus the research in a region that I knew had a ton of sex differences that were really super easily quantifiable. So I choose to look at a region called the preoptic area. The preoptic area has nothing to do with vision, despite the name. It's just called the preoptic area because of where it's located in the brain. It has nothing to do with vision, but it has everything to do with male sex behavior. Masculinization of the preoptic area causes an increase in the number of connections between neurons. So we see more connections between neurons in the male preoptic area compared to the female preoptic area. And if we expose a female to hormones during the critical period, we masculinize the density of the connections. And what's super important about these connections is that they enable the expression of male sexual behavior. So if you prevent these connections from occurring in the male brain, the males can't perform, or they have no motivation to perform. So the first thing I did was I measured DNA methylation and the activity of the enzymes that methylate DNA in the preoptic area. What I found was that during the critical period, females have higher levels of DNMT activity and higher levels of DNA methylation compared to males. So remember that high levels of DNMT activity and high levels of DNA methylation are typically associated with genes being turned off. So in order to determine if this sex difference is actually programmed by exposure to hormones during the critical period, we treated females with a masculinizing dose of hormones during the critical period. And we saw that it reduced DNA methylation and DNMT activity to male-like levels. So this indicated to us that this, in fact, might be a mechanism for how hormones permanently masculinize the preoptic area. But what's important to tell you about is that if we give hormones outside of the critical period, we don't see any changes in DNMT activity or DNA methylation. And this is important because, you know, sometimes you gotta take hormones. Like, you don't want your brain like flip-flopping if you gotta take birth control pills or something like that. So there's something happening during the critical period that allows hormones to control the activity of DNMT enzymes. We don't know what that is yet, but that's something that we're very interested in. So I've told you that females have high levels of DNMT activity, high levels of DNA methylation, and we predicted that the reason for this is because females want to keep tight control over the expression of their genes. They want to silence genes that could be involved in masculinization. And we predicted that this gene silencing in the female brain is what enables brain feminization. In males, who, remember, they have really high levels of hormones in their brains because of this testosterone surge during the critical period, males have low levels of DNMT activity and methylation, and so we predicted that this was causing a disinhibition of genes that are necessary for masculinization of the brain. So in order to determine if this hypothesis is correct, what we did was we wanted to artificially decrease the activity of DNMT enzymes in the female brain. And we predicted that this would lead to male-like patterns of gene expression, male-like anatomy in the preoptic area, and male behavior. And in these studies, we're not manipulating hormones. We already know that hormones cause brain masculinization during the critical period. Instead, we want to know if DNA methylation is a mechanism for how hormones permanently masculinize the brain. So we're just cutting to the chase and we're manipulating DNA methylation. And we did these experiments several different ways. We did them in mice and rats using drugs and other manipulations. And I'm just going to tell you about the coolest one, in my opinion. So we used a virus very similar to the common cold. And this virus had all of the bad viral genes deleted, but it had a few extra genes added to its genome. It had a modification made to its genome that if it infected a cell, it would delete the DNMT enzymes in that cell. And in addition, it had an extra protein called GFP, or green fluorescent protein. And this is a protein that's derived from bioluminescent jellyfish. We predicted that injecting this virus into the preoptic area of female mice would masculinize these mice. And we predicted that, since we have this green fluorescent protein, we'd be able to actually see the neurons that are masculinized. This experiment worked, otherwise I probably wouldn't be up here. So on this side, you see two female mice in a cage. One of them was injected with a control virus. So this virus didn't have the modification that deletes DNMT enzymes, but it had some other stuff in there. And then here on this side, you see two female mice, one which was treated with the real virus that deletes the DNMT enzymes. We could barely start the cameras in time to catch these mice starting to perform male sex behavior. They were so motivated, they basically just ran to the other female and just stayed on top of that female through the entire half-an-hour duration of the test. And 100% of the mice that were injected with this virus performed male sexual behavior. This tells us that high levels of DNMT activity and high levels of DNA methylation in the female preoptic area seems to be necessary for brain feminization, because if we manipulate these DNMT enzymes and we lower their activity, we see masculinized patterns of gene expression. So there's just one last experiment that I want to tell you about. And so far, everything that I've shown you has been manipulations that are done during the critical period, when we know the brain is sensitive to masculinization. But the endpoints that I've shown you, such as behavior and structural stuff, which actually I didn't show you, but you can just take my word for it, that's in adulthood. So the fact that sex-specific epigenetic patterns are established during development, but then maintained to adulthood, made us reason that maybe we could somehow mess up DNA methylation patterns even outside the critical period and cause a female brain to revert to a male brain. So this is what we did. So we administered a DNMT inhibitor to female rats after the critical period had already closed. And this drug is actually a cancer drug that's used to treat tumors, but we injected it directly into their brain, and it causes DNA demethylation. And what we predicted was that this would cause a female brain to resemble a male brain and also allow for females to perform male sexual behavior. On the top row, you see females that were treated outside of the critical period with a DNA demethylating drug. And on the bottom, you see a couple of males. And the females performed male sexual behavior just as well as normal males did. And so, what does this actually mean? First, I think it means that feminization of the brain is actually an active process. Because what I've shown you is that if we take away DNA methylation, even after the brain sex has been established during development, that brain becomes a male brain. So feminization is essentially, at least in the preoptic area, is essentially maintained by DNA methylation. And so it's an active suppression of masculinization via epigenetic processes. It also means, I think, at least in the rat, that the circuitry for male and female behavior is present in the brain, which is an interesting thought, and I think that's different from what we typically believe. And it also shows us that sex differences in the brain are not really as hardwired as we once thought that they were. So we used to think that changes that occurred during the critical period were completely permanent. But the fact that we know now that they're controlled by epigenetic mechanisms indicates that they are a little bit more plastic than we once believed, because we know that epigenetic mechanisms are not necessarily permanent. So in my current work, I am trying to figure out if sex differences in epigenetic mechanisms cause sex-specific vulnerabilities for certain neuropsychiatric disorders. So as I mentioned earlier in the talk, boys are at a higher risk for neurodevelopmental disorders. And could this be that females, since they have higher levels of epigenetic control over their genes, could they be protected from developmental insults that might create these disorders of the brain? So this is something that I'm interested in now, and I don't have an answer for you, but hopefully one day. So that's it. Thank you all very much for your attention.